final say, but I'm sure you'll all do great. You're very good students and very hardworking.
So I'm now waiting for Matt or should
I think we should just start. Yeah.
Okay. So before I start, I wanted to share my experience preparing for this chapter. This was my first chapter summary, I would say. And I've never done this before.
Okay.
But then what I realized was teaching is really difficult.
Well, listen, I mean, in addition to you helping me out, I do think that our graduate students need to do more teaching. I personally feel hi Matt.
More.
When I was a graduate student, I actually taught the methods course by myself.
Right.
I taught that, and I taught another course by myself. There was a professor who was in charge of the course, but they didn't do anything. So I feel that that's an important experience to have, to understand what goes into teaching and how much preparation and all that stuff. Yeah.
And I haven't even started with the presentation. I already feel it because
Right?
going through the chapter, it's not just reading stuff, but also trying to corroborate information from other different sources, trying to make sure what you understand from the chapter is actually the fact.
Yeah.
Is actually happening,
Yes.
and it's not something that is subjective. It is something putative, everyone knows it, but I'm not misguiding or misleading someone by what I understand from this.
Yes, yeah, and it could have been something that you're an expertise in, or even pretty well, but to teach it is a whole different thing.
Right.
So I appreciate you playing along with mine.
And this has definitely kind of changed the way I look at this class, because I now know when you talk about pathways, how much you should be knowing, and how detailed you should be, because everything is so complex. If you go into the nitty-gritties, they're all so well connected, and nothing is black and white. Okay, calcium channel works only by this route. No, there are other stuffs that play in background, which we never know, because we just read about voltage gated channels. We just focus more on some of the key channels and totally forget about the other channels that play a role in repleting or depleting calcium stores.
And then when you're teaching talented students such as yourselves, and you get a question from one of you, and you're like, it's just you spend so much time preparing, and you're like, okay, I'm ready to go, and you get one question, it's just, I didn't think of that aspect of it, and anyway.
So sorry about taking time and sharing my experience, but this was important.
No, yes, I appreciate you relaying that.
So today I'll be covering chapter 7. I'll try to cover within, I'll try to leave some time for Young as well, definitely, because it was a big chapter again. And I'll be speaking about intracellular calcium. When I say intracellular calcium, calcium concentration within the cells, not something. We're not looking at calcium elsewhere, in the extracellular matrix or in the blood. We're looking at calcium within the cell, and that again tells how difficult it is to even gauge where calcium is present within the cell, how much it is present, and especially when the cells have this tendency of maintaining low calcium levels in the cytosol. You cannot say where it is going to release calcium, what pockets of the cell will have calcium, and that kind of adds to the complexity of measuring or sensing calcium within the cells. So though we're not there yet, but the field has advanced so much that we do have tools today that can help sense or at least sense calcium within the cell. And also we've also reached to a point where we can at least, if not very accurately, but then to some extent we're able to measure calcium within the cells. So I'll be talking about intracellular calcium. Long will be taking up the calcium effectors. So on my first slide, and these slides are mostly for me, to what I want to speak, because it was so much that I just had to make points for my recollection and not for the audience, but it's for me more. So calcium, as you know, like is an abundant element. Like most 3% of the Earth's crust has calcium, and it was not before a few experiments that were conducted. We knew that calcium is also a secondary messenger. Earlier, it was thought that, like, despite calcium being so abundant in the cells, it was thought that cyclic AMP is the only important secondary messenger. So cyclic AMP was the first secondary messenger that was discovered. But then, of late, scientists also started studying calcium because of few experiments. For example, in frogs' sartorius muscle, when they removed calcium from the when they tried removing calcium from the extracellular space and also from the cells, it led to blockade of transmission at the neuromuscular junction. So sartorius muscle is basically the muscles starting from the hip to the knees, and in frogs that is governed by the nerve terminals that release acetylcholine. So calcium is responsible for several biochemical functions in the cells, and these include, for example, secretion, then also fertilization, cell proliferation, and most of the you also know most of the excitable cells like neurons, muscles, they have like voltage-gated calcium channels because the entire depolarization and repolarization of these cells depend on the electrochemical gradient that is generated because of sodium, but calcium also has a role to play. It initiates that depolarization.
If you're interested about how muscles contract at the neuromuscular junction and why calcium's involved, he's your man. Because we talk about it in physiology and pathology. Yeah, it's like one of the first things I teach them for the neuroscience section.
So yeah, so most of the biochemical events do require calcium, and it was seen that when calcium was excluded, these biochemical reactions they either stopped or reduced in their intensity a little. So they have the scientific community slowly started realizing that calcium is also an important secondary messenger alongside cyclic AMP. So how it evolved? Initially, the chapter explains this really nice. If you have to see how the importance of how these cell organelles were, they evolved. Basically, you'll have to stage them across different periods. For example, into prokaryotic cells, and then early eukaryotic cells, and then late eukaryotic cells, like in the humans. So humans definitely is a more advanced cell type, but then if you look at prokaryotes, they try to maintain. So every cell will try to maintain low levels of calcium within it, because excess calcium is also it causes cytotoxicity. So every cell will try to maintain low levels of calcium, and how do they do it? They have these efflux pumps or mechanisms in the plasma membrane that kind of effluxes calcium out of the cell. Prokaryotes in prokaryotes they found it, and the very early eukaryotes, like the example that I am showing it over here, the thiovillum sulfur oxidizing bacteria, they were at the intersection of prokaryotes and eukaryotes. And what we see, these linings that you see, this is the beginning of cell organelle formations. These cells started showing some endoplasmic reticulum-like structures within the cells. So, as you know, endoplasmic reticulum in general in the cells, and sarcoplasmic reticulum in the muscles, they store intercellular calcium. So this made scientists think that possibly as the cells evolved, the plasma membranes invaginated into the cytosol, and the calcium efflux mechanisms that they had on the plasma membrane, they actually kind of reversed inside the cell, and some left in the plasma membrane, somewhere in the somewhere on the cell organelles. So as you can imagine, like since it invaginated, the pumps kind of reversed inside the organelles, and on the plasma membrane they still stay intact. So calcium on the plasma membrane, they throw out calcium outside, and within the cells the organelles take in calcium within the lumen basically. So the net result is cytosolic calcium levels are like kept low. They're kept low in the prokaryotes, and now we have like mounting evidence that shows even the eukaryotes they try to keep the cytosolic calcium low. So I wanted to somehow give it some way where you can measure how important calcium is, because it is not just calcium but also magnesium, which is abundant in the cells, and like calcium, magnesium is also a divalent metal ion. So one would think, why just calcium and why not magnesium is important secondary messenger. So there are two reasons. One is the abundance of calcium over magnesium. Calcium is abundant and it is very accessible within the cells. Secondly, the coordination chemistry of calcium versus magnesium. So if you see, magnesium forms complexes that are octahedral, and calcium, they are quite flexible when it comes to forming complexes. And within the cells, they're not just small anions, and they are like protein anions, like big protein anions, which requires more flexibility. Octahedral structure is more restricting to them. So proteins and other biological materials, they do not form complexes so easily with magnesium, given the strict requirement of this octahedral structure. And therefore, if I have to give you an equation, here M is the metal ion, so it can be calcium or magnesium, and L is the coordinating ligand. It can be proteins or for example, for calcium, calmodulin is another protein, and likewise EGTA, which kind of it forms bonds with the divalent atoms. They're like smaller anions, so they equally form complexes with calcium and magnesium, so they readily form it. But then what if you have a long chain peptide with an anion at the end? Magnesium won't be able to form as stable complex as the calcium. So over here in the reaction, Ml is basically the complex, and calcium appears to have higher K value. Here K is basically the measure of how stable the complex is. So if the complex is readily reversing to the dissociated form, M plus L form, then I won't say the K is high. K is high when you see greater partial concentration of Ml, like the complex. So calcium with, say for example, calmodulin forms a stronger complex compared to magnesium with calmodulin. So higher the K value, higher is the stability of the complex. So these two properties of calcium make calcium a more basically a better fit to act as the intracellular secondary messenger. Another property of calcium is its ability to displace water. So within the cell, you don't see ions present free. There are definitely water molecules surrounding these ions, and these are called as coordinating ligands. So the ability to form a stronger complex Ml is achieved only when you're able to displace water molecules. Calcium is bigger compared to magnesium. They can accommodate water as well as the other ligands. So they displace water really well. Once they start forming protein complexes with protein or with other ligands, they kind of displace water really easily. So these are few points which makes calcium a better secondary messenger. Now, Within the cell, calcium is present in three forms. It depends what type of cell it is. So it can be an osteocyte. Like an osteocyte is present as a different. It is present as a trapped calcium, basically within the bones and in the teeth. You see calcium not as iron, but as a mineral. So calcium along with phosphorus is present within the bones and the teeth, and we call them trapped calcium. Trapped because they also act as reservoirs. When, say, for example, in the blood plasma, if you have a hypocalcemic environment, then the bones and the teeth, they come to rescue. They start delivering those excess calcium to the blood. And then you also have bound calcium, like I've been talking since the beginning. Calcium can form complexes with proteins and other ligands. So you also see proteins, which is again another important thing with calcium, which I'll be explaining later. Why it is important. Calcium should be bound to. Why it is important for calcium to bind to proteins. And then you have free calcium. Free calcium, as in the form of calcium ions. Mostly what we've seen is calcium ions, majorly they're present within these intracellular stores like endoplasmic reticulum and sarcoplasmic reticulum, but these are not the only stores. We also have mitochondria. Mitochondria also has calcium in it. And what I would like to, like the idea over here, when I'm trying to explain this table, the idea is to appreciate the fact that how intercellular calcium is maintained low. In the plasma, you see 1 to 2 millimolars of calcium, while in the intercellular lumen, like intercellular lumen as in within the organelles, you see 30 to 300 micromolars of calcium. But if you see in the cytosol, it is just 50 to 100 nanomolar, and that too, not everything is like free calcium. Some of it is like bound, some of it is trapped calcium. So, like I keep telling this, calcium is supposed to, it must be kept low within the cytosol, but how is it kept low? So there are 3 different mechanisms, and probably more than 3, but these 3 mechanisms the book kind of explains really well. So the only way you can keep cytosol concentration of calcium low is either you let the calcium go out of the cell, or allow calcium stores within the cell to take up calcium from the cytosol. So membrane pumps, when I say membrane pumps, I'm referring to the plasma membrane pumps. So cell has a plasma membrane, it has multiple pumps, and these are active pumps. When I say ATP dependent, I also say they're actively kind of, the calcium within the cell, if it rises, they're actively thrown out of the cell using these ATP pumps. There are also ion exchangers, like in the membrane. These ion exchangers, I remember like last time when we were studying advanced pharmacology, neural pharmacology, we did read about 2 moles of sodium accepted by the cell so as to efflux 1 mole of calcium out during depolarization. It is this ion exchanger. So you accept another ion in return of calcium going out of the cell, or calcium going into the endoplasmic reticulum. And then there are calcium buffers. So this is where I would like to say protein binding of calcium is important. So sometimes when the voltage gated channels are open, there is a wave of calcium entering. Though the voltage gated channels open briefly, but then the amount of calcium that enters the cell with the voltage gated channel is higher than any other calcium channel. So somehow the cell has to maintain this balance. Like it cannot exceed a certain, the calcium ions cannot exceed a certain level in the cell. So what proteins do, they actively bind to calcium, and the calcium ions are no longer like available as free calcium within the cytosol. So they act as a buffer. Basically, it's like pH buffers. If you add something acidic, it tries to maintain the pH. For example, if it is 8, it'll try to maintain an alkaline pH of 8. Likewise, calcium here, like an increase in calcium, proteins bind to it and try to maintain some optimum calcium levels. So for example, I'll just like to, I wanted to give it a number to this effect. Calreticulin is a protein that binds to calcium, and around like 1 mole of calreticulin is capable of binding 20 moles of calcium, which is huge.
It's amazing, right?
Right. I know this is slightly dense, but I like to walk you like slowly because this is important. I would have like skipped this out, but then the thing is, really need to appreciate how calcium sensing or measurement of intercellular calcium evolved over a period of time. So earlier, when people realized that calcium is an important secondary messenger, they wanted to quantify calcium within the cells. Like within the cells, how can you best quantify it? Alizarin sulfonate was best known to precipitate in the presence of calcium or any divalent atom. So they tried using alizarin, like injecting alizarin to the cells, or just maintaining cells in the alizarin environment to see if within the cells you see metal ion alizarin complex deposition. It worked, and for a long time people were relying on this method to see the intercellular calcium, but this is not quantitative. The method was not quantitative. There is no way we can measure calcium. It just tells us qualitatively that calcium is present, and not just that. This method was not specific. What if it was not calcium but magnesium and it's precipitating, because alizarin can precipitate out in the presence of any divalent or trivalent metal ions. So then, for quite long, people did not have any tool to measure intercellular calcium concentration. So what they did, like they started using calcium isotopes. So calcium usually is not 45, we know that, but then they have this isotope calcium, which is 45, and the radioisotope can be easily detected using different techniques of basically posimetry can be used to detect calcium isotopes. So they for a quite long period of time they relied on calcium isotopes to measure intercellular calcium, but this was not very accurate. So later what happened was few scientists, they saw within the blue luminescent jellyfish, like occurring from Ecola and Obelia. So these were jellyfishes, marine jellyfishes that had this luminescent material within them. So it luminesces only in the presence of calcium. So they're calcium sensitive, and again, the method was not very accurately quantitative, but still we could sense calcium up to 100 micromolars per liter. And finally, the discovery of, I would say, the invention of fluorescent calcium indicators was very helpful, because now we are able to quantify intercellular calciums really well, if not like accurately, but then really precisely we are able to calculate the intercellular calcium. Two of them include like Quin2 and Fura2. Both of them are fluorescent calcium indicators. So these fluorescent calcium indicators basically the fluorescent properties change when calcium concentrations change. However, I would say Fura2 was more superior to Quin2. There's one reason why, because Quin2 did not provide a calibrated reading like Fura2 did. There's a reason. Fura2 provided progressive wavelength shift with calcium concentration. So on this image over here, like if you see, as the concentration increased, as the concentration of calcium increased, the excitation wavelength that was required to excite the fluorescent material kind of decreased. So you see the shift in the wavelengths that's happening with calcium ion concentration. And again, another thing is increase in calcium concentration also increase the amplitude of emission intensity. So on the x-axis, on the y-axis you have emission intensity, on the x-axis you have excitation wavelength. Excitation wavelength is the wavelength at which the fluorescent material excites. And then when they come back from the higher energy level state to lower energy states, they emit lights. So what they saw, the emission spectrum did not change, but the intensity with which they emitted, like the intensity with which the fluorescent material emitted, increased with the increase in concentration. So this is really important because you can now calibrate. Like say for example, you can take two known concentrations of calcium and then just calibrate against this plot, and then for any unknowns you can just extrapolate the data from.
Like a standard curve.
Yes.
Is it very accurate? I mean, so say you had a standard curve, but your wave was like in between two of those waves. What would you, like I can't see the numbers on, what do they say, 0, 17?
It's 0 nanomolar and then 17. So this is basal level of calcium where they did not add any calcium.
Right.
Then 17 nanomolar calcium, 38 likewise up to like 1.36 micromolar.
Right. So then what if your experiment gave you a line like in between the 0 and 17? All you could say is it's something in between 0 and 17, correct?
So it is more about the peak.
Oh.
So if you see the peak shifting.
Yeah. Okay.
So with high concentration the peak shifts. But at one point the peak remains like the excitation wavelength remains same, and that is the reason like they're able to just quantify like 50 nanomolar to 1 micromolar per liter of calcium. So yeah, Fura 2 is better than Queen 2. And I missed one point over here in calcium sensitive photoproteins. So how do they are different from fluorescent proteins. So they'll fluoresce only when calcium causes some conformational changes in their molecule, in the luminescent molecule. So they are usually not luminescent, but when calcium is present, there is a conformational change. This conformational change oxidizes their luminescent molecule, which is luminescent. So they then show the blue luminescent. So next important thing is okay. Now we have the tools for monitoring calcium within the cells. How do we do that? You can either choose to quantify calcium in a cuvette having cells in suspension. Again, that would give you results like a cumulative result of each cell, and you may not see like single cell effect. So over here, like if I the blue lines that you see is in the like you're monitoring calcium in the presence of extracellular calcium, and the red plot here is monitoring intracellular calcium in the absence of extracellular calcium. So EGTA basically is I keep forgetting this term. Chelator. Yeah, they're calcium chelators. Yes, so they kind of just take up the extracellular calcium and keep your medium devoid of more calcium in the extracellular space. So the basal level did not change irrespective of having an extracellular calcium or EGTA. The basal level did not change. But when we added carbachol at one point, carbachol again is a cholinergic agent which is used as mitotic, and also carbachol is known to increase secretion, like gastric acid secretion. So when they added like 1 millimolar per liter of carbachol, what they saw, there was a spike in calcium levels within the cells. But again, I'd like to remind you, this spike was seen as a whole, like in multiple cells, kind of a suspension of cell was used, and so that is why you're not seeing individual cell level calciums, but then you're seeing as a composition. And then after the carbacols, so these calcium channels do not open, throughout. They open, and then there are some negative feedback mechanisms that cause them to again close. So they open very briefly. When you see like the concentration of intracellular calcium in the presence of extracellular calcium, it kind of plateaus, like it plateaus at levels above the baseline. This is possibly because the extracellular calcium leaking into the cells, and because of several other pumps that are permeable to extracellular calcium. But with EGTA, where there is no calcium in the extracellular space, you see it coming back to the baseline very quickly. This would otherwise, again, this would also come to the baseline, but it takes time. And then next is, how do you measure intracellular calcium concentration at the level of single cell? So they did it using fluorescent microscopy, and they saw over here, what I'm showing you is the effect of histamine, different concentrations of histamine on the single cell intracellular calcium concentration, and the cells that they used was human endothelial cells. And the arrow bar that you see is the time period when histamine was administered. So when the concentration was as low as like 0.1 micromole per liter, you just see a small peak and then it dies out. So this shows that at lower histamine concentration, calcium channels opened, but they opened only briefly. But if you see, as the concentration increased, you see more oscillatory peaks. So they're not similar to like multiple cells in Kuwait. They're not giving responses similar to multiple cells in Kuwait. This is because in single cells there is always waves of calcium, and then you see the declining wave, and then again there is a wave of calcium and then declining. So at lower concentrations there are multiple peaks, but they're separated nicely. They're like nicely separated. As you increase the concentration, the distance between the two peaks keeps decreasing, and ultimately it broadens. Like you see just one peak, and it is a broad peak. This just shows that the calcium channels at higher concentration of histamine, they were open for a longer period of time compared to calcium channels opened at like lower concentration of histamine. Yeah. This is another important thing and a challenge to people who are trying to measure intracellular calcium. It is important to measure the intracellular calcium. It is important that the calcium must be uniformly present within the cell. So, but however, it is very rare that the calcium ions within the cell, they are uniformly present throughout the cell. Usually what happens is the calcium ions are higher close to the calcium channels or at the site of effectors. They cannot be high throughout the cell. So, for example, at the triad region where you see voltage gated calcium channels, immediately next to the voltage gated channel, you will see a warmer region, basically regions like this. You'll see a warmer region, and then more diffuse region away from the channels. But again, at the effector regions, for example, at the presynaptic terminal where you want the neurotransmitters to release, the calcium ions are very close to synaptosomes because you want the calcium to cause that release of neurotransmitter in the presynaptic. But they're not. The calcium levels are not high throughout the neurons. They're just in pockets. They are high. So the idea is I'm trying to explain the micro domains of calcium within the cells. Calcium is not present throughout. It is available in pockets and where it is required, which is really interesting. Mm-hmm. And here what I'm trying to explain is calcium waves in megakaryocyte when they were stimulated with collagen. If you see, earlier it was cool. When I say cool, they're all pseudo colors. So earlier it was cool, as in the calcium release wasn't happening. But now when you see it starts from here, at 50 seconds you see calcium increase in the cell, and by the end of like say 90 seconds, it has become more warmer. That means the cells now, the waves are now traverse across the cells. Likewise, similarly, the similar waves were seen in astrocytes when they were stimulated with ATP. You see pockets of high density calcium regions in the astrocytes.
It's cool.
So now coming to the mechanism, and like very popular mechanism, this is what we have been studying quite long, the IP3-DAG pathway, where calcium channel, intracellular calcium stores are made to release calcium using this channel. So the ligand here is mostly, say, any ligand that binds to receptor and can stimulate the G protein coupled receptor. The beta gamma form of it kind of dissociates and it activates the PLC, which is phospholipase C. Phospholipase C causes the diacylglycerol, which is embedded in these are lipids embedded in the plasma membrane. The phospholipase C causes the inositol 4,5 bisphosphate to break down and gives the byproduct that is formed as the diacylglycerol, which stays in the plasma membrane, and IP3, which kind of enters the cytosol. IP3 here is the ligand that will help that will promote or that will cause the endoplasmic reticulum and the cytoplasmic reticulum to release intercellular calcium. So IP3 binds to the IP3 receptors, or it can be other receptors like the ryanodine receptors, which I'm explaining shortly. These receptors, when they bind to the ligands, they show some conformational changes, and the intercellular calcium then flows from higher concentration region to the low concentration region in the cytosol. So we always talked about IP3 receptors. We never talked about the ryanodine receptors. But then these are again possibly the reason we did not talk much about ryanodine receptors, because they are limited to skeletal cells, skeletal muscle cells, and to the excitable cells like the neurons. But IP3 receptors were like widely expressed across different cell types. Both these receptors, IP3 and ryanodine receptors, they are small molecular weight. Of course, ryanodine appears to be heavier than the IP3 receptor, but the basic construction of tetramers is same. So the reason why they form tetramers is it has to look like channel, like it has to form a channel. So they have tetramers, be it homotetramer or heterotetramer, they form tetramers. And while I was reading this chapter throughout, I saw most of these channels are tetramers, and that makes sense because they form channels, and that's why you need like a tetramer. And both have they're similar structurally. They have 3 isoforms, both of them, and for IP3 receptors, they're present mostly in the cerebellar tissues in the neurons, and type 2 and type 3 types of IP3 receptors, they are widely expressed. Ryanodine has got proper distribution across different types of cells. If you see, ryanodine isoform 1 is present in the skeletal muscles, or I would say more voluntary muscles. Ryanodine 2 has its presence in more involuntary muscles like the cardiac muscles, smooth muscles, and then ryanodine 3 is present mostly in the brain. And I don't want to these receptors. I just wanted to clarify that these receptors are not present in the plasma membrane. They're present on the intercellular stores like the sarcoplasmic endothelium or sarcoplasmic endothelium or the endoplasmic reticulum. And I also wanted to speak about the ligands. For IP3, it is IP3. And other calcium or calcium associated with calmodulin types of ligands. Ligands in the ryanodine receptors are usually calcium, calmodulin, or ATP.
Do all the ligands open the channels, or do some of them close the channels? Do you know?
So it again depends, Dr. Kamna. What I read, these channels have two binding sites. One is low affinity calcium binding sites and high affinity calcium binding sites. When the cells are in their resting stage, the calcium bind with high affinity to the high affinity binding site, and these ligands, they are then responsible for opening the channel. But when they sense that the store is getting depleted, they try to bind to the low affinity, which is more regulatory.
Right.
They have more regulatory.
Which makes
function.
sense. The more calcium that's released from the ER or SR, then once the high affinity sites are occupied, the low affinity sites will then become occupied. So that's a signal to the cell to start closing those channels because there's enough cytosolic calcium. And as you said nicely several times, we have to control that concentration of cytosolic calcium because calcium is such an important signaling protein, and it has to be regulated. Otherwise, the cell would go awry. Right. Yeah.
So I just wanted to, nothing much here, but then just wanted to show you how scientists kind of 3D on 3D kind of reconstructed the rhinodine and the IP3 receptors. So both look similar. They have 20% of their portion submerged in the cell organelle membrane, and the rest 80 or 85% of it like outside in the cytosol. And the part that is in the cytosol both has like C and N terminals there in the cytosol, and the part exposed to the cytosol has these calcium binding sites or the ligand binding sites. So this part basically is submerged in the. So this is side view, this is the top view. Likewise, top view, the bottom view, like from below the channel.
Okay.
The pink side, the pink thing that you see is the submerged
is inside.
portion.
Neat.
Right. So now we know the mechanism of after knowing the mechanism with which intracellular stores open calcium, release calcium into the cytosol. It is just initiation. Like it is just 10% of calcium in the cytosol. We need like more calcium for effective functions. So what happens is when calcium is released into the cytosol, there are other channels that are not associated with rhinodine or IP3 receptors. But as calcium increases, there are these channels that sense increase in calcium, and then they amplify the response. Basically, they start opening, becoming more permeable to calcium from the intracellular stores, and also how do I explain this? So you saw rhinodine 1 receptors, right? Rhinodine 1 receptor is mostly in the skeletal muscles. So the rhinodine 1 receptor it is present in the sarcoplasmic reticulum, which is very close to the plasma membrane. They're in very close position to the plasma membrane, and the channel releases, since they're very close to the plasma membrane, the channel releases calcium in just small microdomain. And what happens is that released calcium then binds to the extracellular binding sites. It's called as dihydropyridine binding sites on the plasma membrane. When it binds to the extracellular channels, dihydropyridine is like interchangeably used with the calcium channels that are present. The name interchangeably used with calcium channels present on the plasma membrane. So when the intracellular calcium, some of it binds to the plasma membrane dihydropyridine sites, the channels on the plasma membrane, they open, and these are called as calcium ion induced or calcium ion induced calcium release, because they open it to the extracellular calcium. The extracellular calcium, they then start flooding into the cell, and this is how like the response is amplified. So
really,
yeah,
that's interesting. I didn't remember that from last time I taught this course. That's very interesting mechanism.
Right.
Yeah.
So as I like mentioned over here, so yeah, Dr. Kavanaugh was talking about this.
Yes.
High and low affinity sites, where low affinity sites are mostly they are into negative feedback, and the rapid increase that we see after initiation of calcium release from the internal storage is because of the calcium induced
So in muscle, skeletal muscle, then calcium binds to a component of the thin filaments, and that's what eventually
Right.
causes the cross bridge. All this must anyway. It doesn't have to. We don't have to go there. But
Right. So yeah, the book also explains twitching of muscles.
yeah. I
There
mean,
are several excitation, contraction, coupling mechanisms.
right. But
uh,
I mean, so then all the calcium that's required for muscle contraction does not come from the intracellular stores. Comes from a combination.
Right. Of
Okay.
extracellular,
Mhm.
intercellular.
Interesting.
And one thing that amazes me is like
I'll tell the PharmD students that, and then they'll hate me for
Yeah.
adding more information. Like what? Anyway, go ahead. Sorry.
one thing that is really amazing is the release is so specific to a particular site. Like it is very close to the channels. It is very close to the effector sites. This is really amazing. Like
Mhm.
you don't see it happening like all over the
Yeah.
cell. It's just happening in
Yeah.
pockets.
If I can interject
Oh,
for
yeah.
the muscle portion,
Yeah.
like this calcium-induced calcium channel is very important for muscle specifically because the calcium release initially that crosses the sarcoplasm into the myofilament. That's to put it very simply, because I don't remember the details of it. The charge is not enough to activate the myofilament, so that's why
It needs a larger
it needs
influx.
larger influx of calcium. So the released amount of calcium induces more calcium release,
Yeah,
and then it kind of reaches that peak, and then
Yeah.
the contraction and excitation process starts.
that's interesting.
Yeah.
Yeah, I taught this material 2 years ago, but since I teach the PharmDs so much, like of 17 years, I keep it simple for them, and we talk about it coming from the sarcoplasmic reticulum. But I might add this. Well, this is actually I'm never teaching that course again, so never mind. But it's an interesting, interesting addition to the knowledge. Yeah.
I think it's also for cardiac muscle as well. Like
Yeah.
this thing is also present in cardiac muscle as well.
It's really common for even neuronal and every excitable cells.
Yeah.
Like most of the excitable cells, they need CICR so that they can quickly mount that requirement of calcium, the required calcium basically. And-
So you mean in neurons, if say IP3 binds to the calcium IP3 channel
Receptors.
receptors on this endoplasmic reticulum, that released calcium then also activates
Yes.
calcium channels on the plasma membrane. How about that?
And here I just wanted to show you again to Afsana's point. There is one more reason. Like we had like calcium as ligand in the ligand section. Like when I was explaining ligands, different ligands, calcium is one of the ligands.
Ah,
That is
yeah,
one reason like we call
right.
them ligands
Right.
because they are inducing other extracellular calcium.
Right.
And on this graph, like I just wanted to show how with concentration, with increase in concentration, the channel opens either briefly or for a long period of time. So at lower concentration, the channel just opens briefly at lower calcium concentration. And at higher concentration, you see the CICR, that is calcium-induced calcium release, is activated.
Yeah.
You see an exponential curve, but then only to a limit you can go where it starts plateauing. But when you increase the concentration, the plateau doesn't shift, but then you see how it's
Yes,
broadening
yes.
at higher concentration. At 10 micromolar, the
That's
peak is broadened.
interesting, right.
That just shows that the calcium channels were they opened not briefly for 10 micromolar, they were open for
It
quite
also
long.
shows that the calcium has to be coming from somewhere else.
Right.
Because the channels are the probability is the same, but there's more
Right.
calcium, so it has to be coming from somewhere else, right?
Right.
Do you see what I'm saying? I think.
Yes.
Yeah.
And also one reason of like when we see fatigue in muscles, you've actually reached the 10 micromolar kind of situation where the calcium channels are like open for like say 1 minute, 2 minutes, and 1 minute like it is like too much for a calcium channel because it is just a fraction of seconds where calcium channels briefly open. They're sufficient to mount the calcium ion in the cell. No, IP3 is something that we keep reading as the only ligand, but then we do have other ligands too, like the cyclic adenosine dinucleotide phosphate ribose is one ligand, and nicotinic acid, adenosine dinucleotide phosphate as another ligands. They're mostly found in pancreas and other types of cells. Like in pancreas, when when the pancreas sends higher glucose levels, what happens is we've also seen cyclic ADPR and NADP increased in that region, in the pancreatic cells, in the beta islet cells basically. And when the researchers kind of tried to exclude cyclic ADPR and NADP, it did not see insulin secretion. So appears like cyclic adenosine dinucleotide phosphatidylose and NADP, they're also involved in calcium, mostly in the secretory functions of calcium.
Look at those molecules.
Yeah, I just added so that because
Yeah, they're crazy. Wow.
and mostly like seen in the like mostly for the secretion function,
Yeah,
and
yeah,
also traffic, like inflammatory cell trafficking, like T cell stimulation.
Yeah.
That we use.
Yeah.
And then another important ligand is sphingosine 1, and the paper that I'll be talking about is sphingosine 1 phosphate, because we have been reading a lot about other ligands, but we never touched at these molecules, which are also important. Like we have compounds in market, like floating in market, that is used for a different treatment like multiple sclerosis, and that's why like I thought of taking a paper that discusses S1B1 receptors and not the conventional IP3DH pathway. So sphingosine sphingosine is actually part of sphingomyelin. Sphingomyelin, these are lipid molecules that line the plasma membrane. They never leave the plasma membrane in sphingomyelin form. But if they leave, and if they leave too much, they kind of act as surfactants and they damage the cell integrity, cell membrane integrity. So that is why even sphingomyelin is kept under like it is strictly regulated, so that not much of sphingomyelin goes into the cytosol as sphingosine or ceramide. So the basic structure is the ceramide structure that we have in the plasma membrane, but the only difference is the side chain. If you see, in the sphingomyelin there is a side chain, and even sphingosine is more hydrophilic compared to sphingomyelin or ceramide. So that is one reason sphingosine can pass through the membranes really well. Another important thing is because of the side chain, sphingomyelin conversion to sphingosine, it is more interchangeable, and that is why they're more likely a regulatory kind of lipids. They very quickly interchange within a fraction of seconds. They quickly interchange from sphingomyelin to sphingosine or sphingosine to sphingomyelin, and that is why they have more regulatory kind of environment. But what is important is sphingosine 1 phosphate, which is an activated sphingosine molecule, which binds at the sphingosine receptors like S1P1 or so sphingosine 1 phosphate binds to multiple receptors, and like there are 5, like S1, P1, P2, P3, P4, P5, but then they bind at like 4 of them: P1, P3, P4, P5. And like when released outside the cell, they're like mostly into cell growth, survival kind of functions, and cell survival functions. Inside the cell, they act as calcium mobilization, act in calcium mobilization from the intracellular stores. And I have the paper that I'll be discussing is targeting the inside of the cell.
In T cells?
In this cerebrocortical terminals, basically neuron terminals. So now we come to the voltage operated calcium channel. I don't think so. Like I need to explain this particular section because this so much, like I would say, an abused chapter in our
Yes.
class. I would say so. Yeah, the channels open. These are voltage sensitive channels. They are available on the plasma membrane, and they are very sensitive to the electrochemical gradient across the cell. So if like there's too much positivity within the cell, they open up. Voltage operated calcium channels, they sense the voltage. Any change in the voltage will lead to an increase in the channel opening. The good thing about this channel is they're very brief. They open for very brief, like fraction of seconds. But then the amount of calcium that these channels are able to mount is like very high compared to any other channel within the cells, like the stores within the cells or any other extracellular pumps. Like even the active pumps, they're not able to mount active calcium pumps basically, or ion exchange pumps. They're not able to mount calcium level anywhere close to what voltage operated calcium channels do. So that is one reason, like voltage gated channels or voltage operated calcium channels are mostly found in the excitable cells, like the neurons or the muscles. And voltage operated calcium channels are like very rare in cardiac muscles, because over there you are more interested in the synchrony rather than it should be more synchronous rather than the amount of calcium that is required for excitation. So the L-type channels that you see, they're mostly present on the skeletal muscles, and the M-type or the PQ or the R and the T-type, they are mostly modulated by the G protein coupled receptor, mostly responsible for intercellular calcium influx. And this I explained previously, that ryanodine 1 receptors, they are in close opposition with the the plasma membrane, and one reason when intercellular stores kind of release calcium ions, they release in that very niche, basically in that space where you have voltage gated channels, and the calcium that is released by intercellular stores act as a ligand on these voltage gated channels to cause more extracellular calcium ion to enter the cell. So if present on the presynaptic neurons, they are mostly for exocytosis functioning, secretion of neurotransmitters. If present in the pancreas and other organs, again they lead to secretion of insulin. And yeah, they're known for the speed with which they open and close, basically open and close. It's brief, and also the intensity with which they cause depolarization is high. So we also have some other calcium channels, not just the voltage gated or the IP3 intracellular channels. We also have some other channels. One of them is like receptor operated, and we know like glutamic acid or glutamate receptors. They glutamate receptors like for example, NMDA or glutamate itself can activate the glutamate receptors, and glutamate receptors, again, they are receptor associated calcium channels. When these receptors are activated, they also cause calcium influx within the postsynaptic neurons. So there's just this two requirement for glutamate or the glutamate receptor associated calcium channel. Number one, the ligand that is binding to the glutamate receptor must activate the glutamate receptors, and second is the membrane must be already depolarized. If the membrane is not depolarized, these receptors are inhibited by magnesium, by the presence of magnesium in it. So if they're depolarized, magnesium is thrown out, and they can then perform this. They can then get activated and cause postsynaptic postsynaptic movement of extracellular calcium to the postsynaptic neuron. And then we have some transient receptor potential. So I know these are like adding to the complexity now. Like it started adding to the complexity, but then they are not widely present. These receptors are not. These channels are not widely present, but then they are present, and it matters when it comes to like an increased glutamate in the circulation, or increased glutamate in the presynaptic space, or an increase in the ADP ribose. If these things increase, at least we have this idea that okay, ADP ribose increase can cause TRPM2 channels to open. So they are again calcium channels. They are basically specific, very specific to say when the cell is under oxidative stress. When they are under oxidative stress, these channels open, and as you know, during oxidative stress, calcium move in and lead to production of more ROS, basically the reactive oxidation species.
Basically kind of saying that the environment's not healthy for the cell,
Right.
right?
Right.
Yeah.
Right. It's more pathological channels.
Yeah,
and-
more pathological, right?
And again, the channels, the ligands that open like transient receptor potential channels are mostly ADP ribose. And this is not the one that I explained before. That was cyclic ADP ribose, and this is ADP ribose. They're different. So ultimately, once the channels open, they start getting depleted. They exhaust calcium since they're releasing calcium from the interstellar stores. So the interstellar stores start getting depleted. Now there are mechanisms in place that help these intercellular stores to replenish their lost calcium, and mostly it is the extracellular calcium that comes back to the intercellular store. And how? So first mechanism that I would like to say is this is a store operated calcium entry. So when the stores start getting depleted, there are sensors, like for example proteins that are expressed or the proteins that TRP gene or the transient receptor potential gene express on the plasma membrane. They have this different transient receptor potential channels embedded in the plasma membrane, and the gene that is responsible for it is the TRP gene. The TRP or the transient receptor potential genes, if they're in the sensory organs, they usually play a role of sensing pH changes, sensing osmotic pressure changes. But otherwise, if they are involved in calcium mobilization, they are mostly involved in replenishing the intracellular stores of calcium. So they are found on the plasma membrane and not on the intercellular stores. They just sense, they are calcium sensors. When the stores deplete in the endoplasmic reticulum or sarcoplasmic reticulum, they come to, like, these genes are activated, and you see more TRPs, TRPC1 to 7 being expressed on the plasma membrane. And that is why this is called store operated calcium entry. So when these stores started getting depleted, it starts expressing this TRP gene. The next mechanism that the chapter explains, this is something that is really new, and I don't know, Dr. Gavna, what year this book was published? Because I call it new, but yeah, even the book says that this is something really new.
Yeah, I think it's 5 years or so
Okay.
old at least. Yeah,
So yeah.
newish. Yeah, yeah.
So it's called calcium release activated inward calcium current. So this is in place already. It's not like the SOCE where the stores one depleted causes TRP expression leading to this is already in place. Like moment the calcium channel opens, even these are activated. So the inward currents are also activated, but then they're slow initially. But in the later part of the calcium dynamic, they kind of become more prominent in restoring the intracellular stores. So calcium release activated inward calcium current. So basically, in the name itself, it suggests that when the calcium is released and the stores are getting depleted, there are some proteins on the plasma membrane and some proteins in the endoplasmic reticulum that interact, and they help refill the intercellular calcium stores. And how do they do it? So when they were studying this mechanism of calcium release activated inward calcium current, they found that a single mutation in this particular gene, ORI1, and that's why ORI1 gene is responsible for ORI1 channels in the plasma membrane. So ORI1 is considered to be a channel. It's a protein which is a tetramer again, and it's a channel on the plasma membrane. Any mutation in the ORI1 developed a SCID type mouse, like severely compromised immunodeficient mice, because this ORI1 expression on the T cells were less on the T lymphocytes. They suffered because of this mutation in the ORI1 gene, and they produced more SCID type of mice. But when the expression of ORI1 was kind of replenished, or kind of when the lymphocytes were forced to express ORI1, we saw restoration of this calcium release activated calcium invert currents, because the T cells were now active. They had more calcium. They had their intercellular stores replenished with calcium, and yeah. So it is the ORI1 channel that replenishes the internal stores. And for ORI1 to be active, like these channels to open, it requires the stromal interaction molecule, which the stim is present on the intercellular stores, like the endoplasmic reticulum or the sarcoplasmic reticulum. You want the stim to cluster, and I'll tell you how it works. So this is the intracellular store, basically calcium store. And in the resting phase, if you see, in the resting phase, you see a lot of calcium. And again, these are stim 1, and stim 1 has this outer portion which is towards the cytosol, and then part of it is submerged in the membrane of the intercellular calcium stores, like the say for example endoplasmic reticulum over here. When the calcium is sufficient, they sense they are like usually bent, and they do not interact with the plasma membrane. This is the plasma membrane. They do not interact with the plasma membrane. When this calcium deplete in the stores, what happens is the outer end of the stim 1, it kind of extends to the plasma membrane, makes connection with the dihydropyridine receptors that I was talking about, which is interchangeably used with the calcium channels in the plasma membrane. They make this connection. It leads to folding of the membrane of the endoplasmic reticulum, and more powerful connections with the channel specific, basically the regions where there are more channels. You will see this fold more prominent. And why this happens is because this interaction with the stim1 and the dihydropyridine receptors, it causes opening of the plasma membrane channels, and calcium is released just in this microdomain, basically. In this microdomain, and not the entire cell. So what happens? This is sarcoplasmic reticulum, endoplasmic reticulum, calcium acceptors, or channels that are present on the intracellular stores. And when this the extracellular calcium enter through because of this interaction, it is present mostly in this microdomain. These calcium can now actively be transported to with the help of SARCA into the endoplasmic reticulum or the sarcoplasmic reticulum. This is how they replenish their stores. So there are three possibilities. Either stim1 interacts with the because this is pretty new. We don't have this idea whether stim1 interacts with the plasma membrane receptors, or stim1 is actively transported in the vesicles to the plasma membrane. And then there is another less known mechanism wherein stim1 is causing release of calcium influx factor, which is less studied. But then there is evidence that stim1 is responsible for release of calcium influx factor as well, which is causing the opening of these channels into these microdomains.
Is that another protein?
Yes, and very less known.
Interesting.
But when they can, in one of the studies, when they produced mice with stim1 knockout, they did not see this calcium influx factor. So somehow it appears like they're more or less interconnected, but then we don't have clear idea as to who is actually responsible, like whether it's the stim1 or the calcium influx factor itself acting independently on the ORI channels. But then what is known is if stim1 is present on the endoplasmic reticulum and there is sufficient ORI1 channels on the plasma membrane, the stores won't get depleted. You will see the stores getting replenished very quickly. So now finally I'd like to share on different channels, on different cell types, how these calcium channels work to activate that. In skeletal muscle, it is usually the voltage-gated channels that play an important role, because you need skeletal muscles are more voluntary, and voluntary functions are quick, rapid. Like for example, slapping someone. Quick,
Example.
That's a good
It's
example.
mostly reflexive.
I'm gonna use that in class.
Yeah.
Actually, I'm gonna go up and just slap someone. How'd that feel?
Was I quick?
Was it quick? Was I quick?
So voltage-gated channels are the first channels that open during a skeletal muscle response, and then how it spreads. Voltage-gated channels, since they open only for a brief period, it needs to spread throughout the cell. And muscle fibers are like long fibers; they're not small fibers. They're not small cells; they're like long fibers. So the ryanodine receptors come to play. When the voltage-gated channels and the calcium enter into these cells, the calcium binds to the calcium binding sites of the ryanodine receptors, causing intracellular calcium to causing the endoplasmic reticulum or the cytoplasmic reticulum to release more calcium, and then the ryanodine receptors that are not attached to the plasma membrane or not in close opposition with the plasma membrane then open after ryanodine 1 actually opens. Because they're not attached to the plasma membrane, they wait for the calcium signals from the ryanodine receptors to open sequentially, and then there is a wave of calcium influx into the cell, which causes skeletal muscles to get excited and contract. So this excitation contraction coupling happens multiple times. If it is a repetitive behavior, like if I'm kind of waving my hands, kind of a repeated excitation contraction coupling that's happening. In the cardiac muscles, behave a little differently, because, as I said, synchronous movements are more important than just the muscle fibers getting activated. That is why over here, what happens is the ryanodine 2 receptors, as I said, are more excessive in the cardiac muscles. But then these ryanodine 2 receptors usually do not have this close opposition with the plasma membrane. So what they do is, the plasma membrane kind of, in this case, invaginates in case of cardiac muscles, and they form close contact with the ryanodine 2 receptors. Over here, I wanted to add that, but I have already added them. So every 10 L types, so when the plasma membrane invaginates and in close proximity to the sarcoplasmic reticulum of cardiac cells, in that invagination, every 10 L type calcium channel of the skeletal muscle is in close contact with, or it engages 100 ryanodine receptors, ryanodine type 2 receptors. So if calcium enters through the L type calcium channel, because of course when the calcium enters through the L type calcium channel, it kind of activates these 100 ryanodine type 2 receptors, and then it does not require any extracellular calcium. It then kind of starts activating the neighboring ryanodine receptors, so that the net outcome is more synchronous activation of each cardiac muscle fibers basically stepwise. So this happens in ventricles. In atrium, it is slightly different, because there's no plasma membrane invagination to the sarcoplasmic reticulum. So it depends on the ventricles to produce basically activate the calcium channels based on ryanodine 2 receptors in the atrium as well. In the neurons, it's like the skeletal muscles again. Voltage-gated channels get activated like the skeletal muscles, but there are also other channels like the glutamate receptor-associated calcium channels present in the neurons too. And in the non-excitable cells, since they're non-excitable, voltage-gated is not the right mechanism here. They're mostly IP3 and ryanodine receptors, so they're mostly internal calcium that is released. They're released in tiny blips, but then, as I said, like ryanodine receptors, not every receptor is meant to give that initial tiny blip. Once there's a calcium spark, and in few hundreds of ryanodine receptors, then it spreads to the neighboring receptors, leading to an overall response in the cell. Okay, so this is the paper that I wanted to discuss today.
By the way, that was excellent. Yeah, it was very good. So we have about 15 minutes, so yeah,
Yep.
okay.
So I won't cover the G protein part of it, like we have had enough of beta gamma, I guess.
Yes.
And so I didn't want to add this to my presentation, but I would rather touch on the S1P1 receptors, which is part of my just to
Fingolimod.
stay.
Do you remember fingolimod?
Fingolimod.
Fingolimod, I'm sorry.
Fingolimod inhibits
We talked about it for
MS.
treatment of MS, yes, thank you.
So yeah, so
Fingolimod, I'm sorry, I always say it right, wrong, I forget the O.
So glutamate, as you know now, that it is responsible for influx of calcium into the synaptic neurons. Glutamate release in the neurons causes excitability, can cause like epileptic seizures, or in case of multiple sclerosis as well, we have seen excitatory response in the neurons are prevalent in multiple sclerosis as well. So this paper actually wanted to check if fingolimod is able to inhibit glutamate release, and they also wanted to derive the mechanisms by which fingolimod does that. So the objective of the paper is to study the effect of fingolimod on the release of glutamate in rat cerebrocortical terminals, like the nerve terminals. So they have taken the presynaptic nerves of the cerebrocortical neurons. So the first finding that I would like to show is like fingolimod indeed reduces the glutamate release in the presynaptic space. In the panel to the left that you see, on the x-axis it's time, across time, what is the glutamate release when fingolimod was given, or when mock fingolimod or control was given. So fingolimod did not change the basal, like the basal response of glutamate release, but when 4-aminopyridine, so 4-aminopyridine here is given to increase the glutamate release. 4-aminopyridine is basically a potassium channel blocker, and as you know, when we block potassium, it's basically the ion exchange. When you block potassium channel blockers, there's an increase in the calcium in the cell, and it causes the cell to become more depolarized. Right? So it is the 4-aminopyridine induced glutamate release at this point, and you see with the control there is an increase in the glutamate release, whereas this increase in the glutamate release is muted by fingolimod in the fingolimod treated group, reaching significance around 400 seconds. And then on the second graph, you see this decrease in the glutamate release is actually dose dependent up until a point like where you have 30 micromolar of fingolimod, but beyond that it had no effect on the glutamate release. So
I forget what we call that, what's that?
Bell shape, inverted bell shape.
No, there's a,
U-shape.
no, there's a word for it. Oh. Because this happens a lot. You get this, like,
U-shape.
just go ahead, I'll come to it. No, there's actually like a word that's, if I don't come to it today, I'll bring it next time.
This paper is really complex, so I had like really hard time understanding the paper, but I'm still trying. I
It's
try my best
fine.
to,
That's good,
explain
that's
this.
alright.
Okay, so they next wanted to check if S1P1 receptor, which is the receptor for sphingosine 1-phosphate.
It's called a hormetic effect. Hormetic or hormetic effect. When you see that, it's a hormesis.
Mm.
That's what I think I'm saying it right. Go ahead.
So they wanted to check if S1P1 receptors are somehow associated in this process of fingolimod inhibiting glutamate release. So I'll just like very patiently try to explain what are these. Like fingolimod is the treatment here. SEW2871 is the S1P1 agonist. Suramine is the S1P3 E5 joint agonist, and W146 is the S1P1 antagonist. DMS, or I think it's I forgot the full form of this, but then it's sphingosine kinase inhibitor. So sphingosine kinase, you know, sphingosine, it converts sphingosine to sphingosine 1-phosphate. It's kinase, so it does phosphorylation of sphingosine. So in the absence of everything, which is control, and on the y-axis you're seeing the glutamate release as the percentage of control itself. So since this is the control arm, you're seeing 100% glutamate release for the control. And then when fingolimod was added, there is a significant decrease in the glutamate release. In the presence of the S1P1 agonist, there was a significant decrease in the. I would consider these two independently because now I'll have to take independent way of explaining. If you look at this and this, you'll say how come S1P1 agonist is kind of reducing the glutamate release. But when S1P1 agonist was added, basically S1P1 S1P1 agonist is blocking the fingolimod receptors. So fingolimod acts on S1P1 receptors. Now S1P1 is blocked by the S1P1 agonist. So it does cause a reduction in the glutamate release. But then in the presence of fingolimod and the agonist, the fingolimod is not able to give us even like more. It is basically abolished the inhibitory response that fingolimod had. It just shows that S1P1 is involved in the in this reduction of glutamate release. Likewise, when we had like S1P3, this is just to check if S1P3 or P5 is also involved. But then when they tried it, like S1P in fact kind of had more or less like a response like a control. But then fingolimod was able to was still able to reduce the glutamate release. So this shows that S1P3, P5 joint is not involved in this mechanism. They also did the Western where they saw the absence of S1P3 receptors itself. And then the S1P1 antagonist, they somehow showed that S1P1 antagonist also reduced the glutamate secretion, and they reasoned it as an increase in antagonist concentration, leading it to become more inverse agonist kind of molecule. So, but then yes, again glutamate could not reduce it further. So that just shows that S1P1 receptors are important. And with sphingosine kinase, when sphingosine kinase was inhibited, it showed response similar to the control, but at the same time, when in the absence of sphingosine kinase, fingolimod could not reduce the glutamate release. This shows that
the kinase is downstream from the receptor.
Yes.
Yeah.
Yes. So it shows that S1P1 is involved, but at the same time, sphingosine kinase is important for fingolimod's activity in effect of reducing glutamate release. And then they wanted to see if glutamate transporters are co-localized with S1P1 receptors. So what they did an IHC study wherein they double labeled for vesicular glutamate transporters on like in the A, and then even synaptophysin is just a control wherein it's a general synaptosome marker. So yeah, they did that, and so they just wanted to check if vesicular glutamate transporter co-localizes with the S1P1 receptor in these cells. So they did see that. Like you see the green, in the red channel they put vglut, basically glutamate transporter, and in the green channel they put S1P1 receptors. And like when they overlaid it, like they just overlapped it. In the overlapped version shows like red and green with yellow spots here. Like you see that they're basically overlapping. So basically they just proved that S1P1 receptors and glutamate transporters they're kind of co-localized in these or co-expressed in these cells. Similarly, this is just a control wherein to see that they're not off-target effects. The synaptophysin and S1P1 receptors were tried to co-localize. They tried to co-localize it, and they did see the same. So basically, what they were looking at was S1P1 receptor and vglut. And again, they wanted to check if this response is calcium dependent or calcium independent. So as to like, is it dependent on the extracellular calcium or not? So CaCl2 was just provided in different concentrations so as to provide an extracellular calcium environment. Fingolimod was given, and then EGTA was added to deprive the cells of the extracellular calcium. And then you have glutamate transporter. Glutamate transporter basically to glutamate transporter transports glutamate outside the cell in the presynaptic space, so that it kind of actually blocked the glutamate reuptake. So yeah, and bephilamycin is basically a vesicular transporter inhibitor. Vesicles which carry the glutamate, if we inhibit that, what happens? So what they found was with with nothing in it, it shows like 100% response, basically because it is like normalized against control. And with fingolimod and the calcium, they did see a depression in the glutamate release. And with EGTA present, they did see a depression, but not to the level of what we saw with fingolimod alone. So this just shows that extracellular calcium was extracellular calcium entry into the presynaptic neurons was essential for the release of glutamate. Yeah. And when they added EGTA, the response was maintained. Basically, it was the same response as the fingolimod and calcium alone. And likewise, when they added, so fingolimod was not able to reduce this response. Like when EGTA was added, though there was some reduction in the glutamate release, because you don't have extracellular calcium. And that is the whole point. Like they're just trying to prove this point that extracellular calcium is essential for the glutamate release.
Yeah.
And again, in the presence of fingolimod, it could not decrease further. That just shows that extracellular calcium entry into the presynaptic neuron is essential for the glutamate release. And when they tried like blocking the glutamate reuptake, they saw they saw an increase in the glutamate. Of course, glutamate was not getting back to the presynaptic neurons, and that is why in the presynaptic space there was too much glutamate. But then this response was kind of reduced by fingolimod. So basically, they were trying to show that reuptake is not an issue. Like, reuptake is not the mechanism that's involved in fingolimod mediated calcium suppression. Glutamate suppression. And then vesicular transport, it is involved because it led to reduction in the glutamate release, but fingolimod could not any further reduce it. So if it is not able to reduce it any further, basically they meant that fingolimod is basically acting by that route of. And finally, they wanted to show that whether extracellular calcium has any effect on fingolimod effect of reducing the glutamate release. So in this, there was a control group, and then we have different agents that they have added. Fingolimod along with N-type CA blockers, PQ type calcium blockers, Dentroline, and this molecule is basically intracellular calcium release inhibitors. So they wanted to check if it is intracellular inhibiting intracellular calcium versus extracellular calcium, like blocking extracellular calcium. So what they saw with fingolimod, again, they saw a significant response, and when they just used the N-type blocker, it did abolish some of the fingolimod response, but fingolimod was able to get back the reduction of glutamate release, like to the level below the below this arm, which just has fingolimod. And then they also checked the PQ type of calcium channel blocker. Again, it saw a similar response, but this time it was far reduced compared to the N-type calcium channel blocker. Fingolimod was not able to reduce this response any further. And when both N-type and PQ type calcium blockers were added together, basically they are both extracellular. They are channels for extracellular calcium. So when both these agents were added. Both N-type and PQ-type calcium channels were blocked. Fingolimod could no longer, reduce it. So basically, that just shows that both N-type and PQ-type are involved in the fingolimod's effect of reducing glutamate. This was not seen with dentroline, which is intracellular calcium release, or with the other reagent which is responsible for inhibiting intracellular calcium. So they just, in summary, what they showed was they kind of tried to, like from their learnings, they tried to map the mechanism with which fingolimod acts. So fingolimod, so this is the part that I did not cover today. Basically, they also showed that it works by G protein coupled S1P1 receptors, and so where G beta gamma is more regulatory kind of thing. It blocks the calcium influx from the N-type or PQ-type channels, and this blocking of voltage-dependent calcium channels kind of reduces the intracellular calcium, and then when the intracellular calcium is low, of course the secretory function of the neurons suffers here. The vesicles are no longer able to fuse with the membrane and release the glutamate into the presynaptic space.
Are they saying that this is one mechanism of action of fingolimod in multiple in MS?
Yes, like one of the different, maybe one possible route with which.
Because we talk about it in class, how it inhibits the efflux of activated T cells from lymphocytes. Fingolimod.
Okay.
Fingolimod.
Yeah.
So this is interesting.
That was more, I guess, with respect to MS, I guess.
Yeah, but this is more for what?
This is more for excitotoxicity.
Excitotoxic, which is also plays a role
Correct,
in
MS. Yes.
Yeah. Interesting. That's very interesting. Because I didn't know about this mechanism, so I'll have to add that to my notes too. Very
Thank you.
good. Thank you. Vivek, you did an excellent job. You will be.
I took like Young's time.
If you
I'm so
ever
sorry.
want to be, another thing I wanted to tell you about this, that since this is technically like a teaching exercise, if any of you graduate students, I don't know, Matt, if you're eligible for it, but they have teaching certificates and teaching awards. If you ever want to go for that, I can use this as observing you teach, and I can write a letter towards that. Okay,
Oh, nice.
so just keep that in mind when that comes up.
Sure.
Okay, I don't.